Hey, hello and welcome back again to another video tutorial of Royce Academy. In this video tutorial, I'm going to discuss on another important aspect of biosignaling, which is basically dealing with the counterbalance or the antagonistic phenomena to that of functioning of the insulin hormone. Okay, in case of insulin hormone functioning, we have seen that how the insulin hormone is functioning to help maintain the normal blood glucose level by cleaning off the excess glucose from the blood in the hyperglycemic condition. And in this video, I'm going to deal with the glucagon aspects. Glucagon is the hormone which is secreted from the alpha cells of the islet of Langerhans of the endocrine gland pancreas, and it helps regulate or upregulate, upregulate the quantity of the glucose during hypoglycemic condition, that is when the glucose molecules are too much less in blood. Okay, so let's have a check that how the glucagon hormone is functioning antagonistically to that of insulin hormone in order to maintain in order to build up, restore the normal blood glucose level. Okay, so when the insulin hormone, when the insulin hormone, it's getting secreted from the beta cells of the islet of Langerhans, right? And now when the glucagon hormones secretes from the alpha cells of the islet of Langerhans via the blood capillaries or the blood vessels, it is translocated or transregulated into the liver cells. Now, what is the function going into the hepatocytic cells? What is the cascading of the glucagon hormone within the hepatocytic cells? So I've drawn a rough diagram of the plasma membrane of the hepatocytic cell. As usual, these two ends, the redhead ends are the polar ends, which are the hydrophilic, and this middle section is the hydrophobic ends on the non-polar end, and this portion is the cytoplasm, and this is the cytoplasm or the intracellular space, and this one is the extracellular matrix or the portion outside the cell. Now, suppose when the glucagon hormone is coming, and first it gets bind with the glucagon receptor present across. Glucagon receptor is a kind of protein molecule which remains attached or latched on across the entire surface of the plasma membrane. Okay, so this is what the glucagon receptor looks like. Okay, so this is the glucagon receptor, and the glucagon hormone would gradually bind with this receptor. Okay, now when this glucagon hormone, this is the glucagon hormone, and this is the glucagon receptor, GR. Okay, and this glucagon receptor has a specific dynamic activity. After it gets the signaling from the glucagon hormone, the glucagon receptor helps in activating a primary messenger molecule, which is itself again a protein molecule that relies within the lower portion, that is towards the intracellular head of the plasma membrane. Let's see what this protein is. The glucagon receptors activate another protein molecule which is a secondary, which is a primary messenger or a secondary protein molecule which is for this performing the signaling aspect within the plasma membrane. This protein is called particularly the GS. GS is actually the one of the subunits or one of the kinds of the G proteins which acts as the primary messenger in order to transmit the biosignal from the glucagon receptor or from the receptor that is present across the plasma membrane. The glucagon hormone itself is acting as the ligand in order to activate the receptor, which is thereby activating the glucagon, activating the primary messenger, the GS protein. Okay, now this GS protein which is getting activated is again activating another enzyme Okay, another messenger molecule which might be called as the tertiary, okay, or a third degree messenger molecule coming under the classes of the primary messengers, which helps in transmitting the signal from the GS subunit or the GS proteins, G proteins. This enzyme is nothing but adenyl. The name of this enzyme is adenyl cyclase. So what is happening here basically is the glucagon hormone, which gets secreted by the alpha cells of the islet of Langerhans of pancreas, are being trans-regulated or transported via blood to the glucagon receptor, which is relying across the plasma membrane of the hepatocytic cell. This is nothing but a hepatocytic cell. Now, once the glucagon receptor is getting activated by receiving the signal from the glucagon ligand, it helps in activating a primary messenger molecule, which is a G protein molecule called GS unit or GS subunit. And this GS subunit in turn helps in activating an enzyme molecule called adenyl cyclase. Thus, adenyl cyclase helps in converting this adenyl cyclase molecule ATP molecule, ATP or adenosine triphosphate molecule, to cyclic adenosine monophosphate, that is CAMP. Now, the ATP molecule, the adenyl cyclase enzyme is converting the ATP to CAMP, which is rather getting activated. Now, this molecule is active form. Okay. Now, this activated CAMP will again help in activating another signaling molecule. The CAMP is here. The CAMP molecule is acting as the secondary messenger molecule. Okay. The CAMP, cyclic adenosine monophosphate, will help in activating another messenger molecule, another secondary messenger, which is called as protein kinase A, PKA molecule. And this protein kinase A will get activated in the presence of CAM or cyclic AMP, which would help in formation of the active protein kinase A molecule. Okay. This inactive form of the protein kinase A by the secondary messenger CAMP, cyclic adenosine monophosphate, is getting activated to the protein kinase A. Now, the function of this protein kinase A molecule is nothing but helping in activation of the phosphorylase kinase enzyme. This activated protein kinase A will again help in activating a particular enzyme which is called phosphorylase kinase, PO4 kinase B, and helps in converting the phosphorylase kinase B to the phosphorylase kinase A, which is an active form, PO4Ka, which is present in the active form. Okay. Now, the function of this phosphorylase kinase A is to transfer the phosphate group to the glycogen phosphorylase enzyme, which is the major enzyme required for, which is the checkmate enzyme, which acts as the bingo in order to perform the last and one and only step, that is glycogen synthesis in case of this glucagon cascading. Okay, without the activation of this enzyme, the major part that we are going to deal with, the storage of the glucose to the glycogen molecule, in case of synthesis, insulin was happening, but in case of the glucagon molecule, the glycogen phosphorylase, that is the breakdown of the glycogen to the glucose molecule, would not happen. So it's a checkmate part. That's the major enzyme which is acting as the bingo in order to perform the one and only vital step of conversion of the glycogen to glucose and elevate the blood glucose level by throwing out the glucose into the bloodstream out of the intracellular space to the extracellular matrix and is being carried out the blood in order to elevate the blood glucose level when the blood glucose level is coming down beyond the threshold value, beyond the normal value. Right. Now this phosphorylase kinase enzyme helps in activating or transferring the phosphorylation, the phosphate group, to that particular enzyme called the glycogen phosphorylase. Glycogen phosphorylase. Now this glycogen phosphorylase enzyme is getting activated. The glycogen phosphorylase enzyme is getting activated with the help of the transfer of a phosphate group here. A phosphate group, because it is getting phosphorylated due to the presence of this active phosphorylase kinase A. This active phosphorylase kinase A helps in activating the glycogen phosphorylase by phosphorylating it. Okay, now this molecule right up here is present in its active form. Okay, so this is nothing but the active form of glycogen phosphorylase. Now what does this glycogen phosphorylase do? This glycogen phosphorylase is the only enzyme which would help in phosphorylating the glycogen polymer. Okay, the glycogen polymer, this is how the glycogen polymer looks like. This is what the glycogen polymer looks like, having the branched alpha 1,4 and alpha 1,6 linkage. Okay, now this glycogen phosphorylase would help in donating the phosphate group to the glucose molecule. Okay, it would help in donating the phosphate group to the glucose subunit or the glucose monomers present within the entire glycogen phosphorylase, glycogen molecule, entire glycogen polymer. And now once the phosphate group is attached with the glycogen polymer with the glucose units, it would help this phosphate group or radical would help in breaking down the glucose molecule, the glycogen polymer to the glucose molecule. Along with the phosphate group. And now this glucose molecule along with the phosphate group in the presence of another enzyme called the phosphatase with the removal of the phosphate group helps in converting the phosphorylated glucose to the simple glucose molecule, that is this one, simple glucose molecule. So this is the intracellular space or the cytoplasmic space, and this is the extracellular space. Now once the phosphorylated glucose broken down from the phosphorylated glycogen polymer, the phosphorylated glucose is with the removal of the phosphate group in the presence of phosphatase enzyme is getting converted to the glucose. The glucose molecule is readily thrown out of the intracellular space or the cytoplasm to the outside of the cell, and from here it is being carried out by the blood vessels or the capillaries or whatever it is, the blood vessels and all, the arteries and veins and all, and circulated to different other parts of body by the veins and capillaries and all. Okay, so this is how the breakdown of the glycogen molecule is happening with the help of the cyclic adenosine monophosphate, which helps in converting the protein kinase A, inactive protein kinase A to the protein kinase A active form. Now how does the activation happens here? You see the CAMP molecule present right here, this cyclic adenosine monophosphate. The ATP, in the presence of adenine cyclase, the ATP is getting converted to CAMP plus an inorganic phosphate molecule. Okay, now this inorganic phosphate molecule would come and bind with the phosphorylase kinase molecule, okay, and help in activating it. Now this phosphorylase kinase will donate this phosphate to this inactive protein kinase A, the protein kinase A, which is phosphorylated, would donate the phosphate group to the inactive phosphorylase kinase B, and hence rather this molecule will again have the phosphate group. Now this phosphorylase kinase A molecule, which is activated, would donate the phosphate group same to the inactive glycogen phosphorylase, and hence it would activate this glycogen phosphorylase molecule. Now this glycogen phosphorylase will again, what it will do, it would activate the glycogen phosphorylation by donating the phosphate group to one of the glucose units of the glycogen chain, which is having the alpha 1,4 and alpha 1,6 linkages, and phosphorylate one of the glucose residues and break down on one of the glucose residues by disturbing this chain, disturbing this alpha 1,4 and alpha 1,6 glycosidic or trans-acid glycosylase bonds, and help in forming the glucose, phosphorylated glucose with 1-phosphate, which in the presence of a phosphatase enzyme which remove the phosphate group and get liberated or thrown out of the cell into the bloodstream. Okay, now this is all about how the glycogen is getting converted to the glucose in time need of emergency when during hypoglycemic condition, when the glucose level have fall down beyond the threshold value, beyond the minimum value. When it is hypoglycemic, that is the glucose levels are not at all present within the blood. Okay, in case of adversive or detrimental conditions. Now suppose this all happens in the case of elevated levels of the glycogen molecule. Elevated levels. Okay, now suppose once the glucose molecules, the normal value of the glucose molecules are being restored, then what happens, the glucagon molecule would have a feedback antagonistic role or signal on itself, which would lower down the secretion of the glucagon from the alpha cells of islet of Langerhans. Now in case of the lower value or lower levels of this glycogen, of this glucagon hormone, what would happen? This glycogen receptor with the lower levels of the glucagon hormone, this down arrow, this down arrow sign means the lower level, and this up arrow sign means the upper level. Okay, so for this particular lower level of the glucagon, low concentrations of glucagon would bind with the glucagon receptor, and the glucagon receptor would then in turn help in activating a particular enzyme. Okay, a particular enzyme via another G protein subunit. That G protein subunit is called the the GP. Here it was GS, and now this protein subunit is GP. And now this GP, which is another protein subunit of or another type of G protein, would help in activating a particular enzyme which is present towards the intracellular head of the plasma membrane. Okay, in this particular portion, in the lower portion or lower end intracellular head of the plasma membrane, this GP protein rather activates another enzyme which is known as phospholipase C. Okay, phospholipase C. Phospholipase C. Now this phospholipase C enzyme would help in the breakdown of or the conversion of another substrate molecule or phosphoglyceraldehyde or phospholipid that is present within the plasma membrane. The phospholipase C would help in breakdown of that molecule. The name of that molecule is phosphatidyl inositol 4,5-bisphosphate. Okay, phosphatidyl inositol 4,5-bisphosphate. It's a phospholipid or a phosphoglyceride. Okay, or it might be called as glycerophospholipid, which is a substrate of this particular enzyme phospholipase C, which helps in splitting that PIP2 into two different molecules. So the name of that particular molecule is PIP2. Now this PIP2 molecule, with the action of this phospholipase 2, would break down to two different molecules. One of them is IP3 or inositol triphosphate, and another one is DAG or diacylglycerol. Now what happens is these IP3 molecule present right over here, this IP3 molecule would have would play a major function in in this feedback inhibitory pathway. This pathway is the synergistic. This pathway is actually the direct pathway, and this pathway is actually the antagonistic pathway of the glucagon itself. It's a feedback inhibition pathway. Why it's called a feedback inhibition pathway? I will show you. This IP3 molecule or inositol triphosphate readily comes and binds with the calcium channels of the endoplasmic reticulum. Okay, suppose this is the endoplasmic reticulum present right over here, and now here lies the calcium channels. Okay, suppose this is an example of the calcium channel present right over here. Okay. Now this is a calcium channel present right over here. So this calcium channel, this IP3 molecule is like a door stopper. Okay, suppose these two ends, you see these two flaps of this calcium channel. Suppose these two flaps acts as the two doors. Another IP3 molecule acts as a door stopper. It will directly come, this molecule will directly come and sit on, or as a weight, as a inhibitor, okay, as a tension molecule, as a tensile strength, or as a what I'll say, like a elastic rubber band kind of appearance, as a hinge. It will sit on, like a door stopper, exactly. It will sit on this particular molecule. Okay, the IP3 molecule will come and sit on the door, sit on one of the flaps of, or sit on one of the channels of this calcium flaps. Now when it sits on one of the flaps of the IP3 molecule is sitting on the on one of the flaps of this calcium channels, one of the covers of one of the doors of the calcium channels present right over here, this IP3 molecule will not help in the closure of the calcium channel. That is in absence of the IP3 molecule, this particular flap present over here and this flap present over here would not close. These two flaps would not come towards each other. Okay, it would not help in the closure of the calcium channel, and as a result, what would happen? Excess calcium ions, it would keep open the calcium channel, and the calcium ions Ca2 plus would come out of the endoplasmic reticulum. The Ca2 plus ions would come out of the endoplasmic reticulum, because endoplasmic reticulum is normally a reservoir or a pool of the calcium double positive ions. And now when the Ca2 plus ion comes out of the endoplasmic reticulum, these Ca2 plus ions, along with this particular molecule diacylglycerol, okay, along with this particular molecule, together, this Ca2 plus ions along with the calcium double positive ions together would help in activating this calcium double positive ions, and the DAG, diacylglycerol, would help in activating another protein molecule, another enzyme, actually another proteinaceous molecule, which is an enzyme called protein kinase C. Okay, protein kinase C, PKC. Now this PKC, in its activated form, as well as this calcium ions, both of this protein kinase C and this calcium ions have a feedback inhibitory function or an antagonistic function on this particular direct pathway. Okay, it helps in shutting down this pathway. Now why it happens? First, let us have a look on the calcium double positive ions. these calcium double positive ions would directly block or stop the pathway of adenyl cyclase activation. Okay, and this protein kinase C, the activated protein kinase C molecule, would help in the phosphorylation. This activated protein kinase C would help in phosphorylating the adenyl cyclase enzyme or adenyl cyclase molecule. Now, on one hand, the calcium double positive ions is negotiating with the adenyl cyclase enzyme. Okay, the calcium double positive ions is it have a synergistic effect as well as an antagonistic effect. It helps regulate the adenyl cyclase enzyme. In case of lower concentrations of the glucagon, okay, it needs to decide that whether this direct pathway needs to be performed in order to elevate the glucose level, in order to break down the glycogen and convert the glycogen to the glucose, or when the glucose molecules have reached its saturation point within the blood glucose level, further the adenyl cyclase activation of this entire direct pathway is not needed. So it would act as an antagonistic. It would inhibit, it would send a feedback to this adenyl cyclase to inhibit or stop the process. Okay, so the lower concentration of glucose is actually the feedback inhibitory pathway. It would inhibit the activation of the adenyl cyclase. And now this activated protein kinase C, which is getting activated in the presence of diacylglycerol and calcium double positive ions, is helping in phosphorylation of these adenyl cyclase. Now, once the adenyl cyclase gets phosphorylated, it would get inactive. Okay, so in both the cases, in the duo, that is in the presence of the calcium double positive ions as well as in the presence of the protein kinase C molecule, in both ways the adenyl cyclase molecule is getting shut down. Okay, now once the adenyl cyclase molecule is shut down, it would automatically shut down this entire pathway. Now, when it shuts down the entire pathway, that is when the normal blood glucose level has reached. So the glucagon hormone does not need to be secreted or does not need to perform anymore in its normal higher concentration in this pathway. Rather, it would perform this pathway, that is a lower concentration, and block the activation or the operation of the adenyl cyclase. Right? So this is how the glucagon hormone is cascading within the cytoplasm of the intracellular matrix, intracellular space of the hepatocyte cell within the liver cell. Okay, so this is what this is how the glucagon hormone is functioning in order to maintain the normal blood glucose level, and via its own feedback inhibition, it is controlling its self-regulatory aspect from the plasma membrane. Right? So if you like this video, positively please do subscribe this channel in order to stay updated with more such videos and all, and comment down in the comment section below, share the video as much as possible and hit the thumbs up button, and also don't forget to let me know if you have any personal queries or doubts. Thank you.